I guess you ought to expect this sort of an outcome when you ask a scientist to explain medical terminology, but speaking of cycles, I noticed that there was some confusion about the role of GTPases in controlling cyclical processes, and I thought I'd try to shore that up a bit. The basic point that I think is missing is that GTP is used as a timer in various cycles in biology. So it's unlike a kinase where we're taking the phosphate from some high energy donor and putting it on another molecule. This is a process where we say, I need this cycle to be activated, but I don't want it to stay activated because that's dangerous. So what I'll do is I'll place a timer on it. I'll put this GTP into the active site of a GTPase, and it's not going to last very long. Then I can turn the process off automatically. I don't have to come along and do something to throw that switch. I can just automatically let it shut itself off, and then I can control how long I'm going to let it stay on by how long it takes me to hydrolyze the GTP. Here in the sort of classic example with the Ras proteins, these small GTPases, we have two forms of Ras. One with GDP bound, which is inactive, that then becomes activated by acquiring GTP, which of course also requires that we get the GDP out of the active site. So we're going to have a GTP exchange factor that's going to be important in the cycle because the GDP doesn't fall out by itself in this case. Once that GDP has been removed, it's easy to get a GTP. Now we've got the active form. That's going to stay active until Ras, which is a GTPase, decides to hydrolyze it. Once it's hydrolyzed, it goes back to the inactive form. So the GTP is acting as a timer in this case. If we look at it in the context of where the cycle works, we've asked you to understand this signal transduction pathway where a receptor tyrosine kinase over here in the cell membrane gets activated by having some mitogen, some small molecule that's going to tell the cell to proliferate, will bind to the receptor tyrosine kinase and it dimerizes. And the thing that it does, its substrate, is going to be through a couple of intermediaries, one of those GTP exchange factors. The GTP exchange factor then gets activated. It takes the inactive Ras with GDP, removes the GDP for it, and lets the Ras become active. If we want to shut this off, we can just wait for Ras to hydrolyze the GTP, or we can actually make that faster by putting in a GTPase activating protein, these gaps down here. So we can control the amount of time that it spends in the active form. While it's in the active form, it goes to the membrane and activates one of our kinases, RAF, at the top of this cascade of kinases. This activation doesn't involve a post-translational modification. It just involves localizing RAF and contacting it and telling it to be active. So this first activation in the cascade is a localization and activation through direct contact. Then downstream of that kinase can activate MEK, which is itself, of course, a kinase that's inactive, by phosphorylating it, and then MEK can activate downstream ERK by phosphorylating it. So the rest of the events in the pathway are phosphorylations by kinases, but that first one is just Ras localizing and contacting RAF. This is the pathway that we've asked you to learn because it's important in lung cancer and because we have drugs that inhibit these various kinases. Ras itself isn't a very good target because we don't have any way to prevent it from doing this activation step because it doesn't involve a catalytic activity. It's important to point out that this is a general class of signal transduction pathways called the MAP kinase pathways. There are various receptor tyrosine kinases and various MAP kinases that are going to be involved in these mitogen-activated protein cascades. They all end up activating a mitogen-activated protein kinase, like ERK, which then goes on and activates transcription of things that are going to promote proliferation. In general, the thing that phosphorylates the MAP kinase is a MAP kinase kinase, and the thing that activates it is a MAP kinase kinase kinase. Great terminology, huh? But that's the general form. The one that we want you to learn is this particular one. But it's important to know that there are dozens of these signal transduction pathways in human cells that regulate proliferation. Tubulin also fits into this same sort of a scheme where we're going to use GTP as a timer. So it's not in the Ras family, but this same process of using GTP as a timer to limit the activation cycle so that it doesn't go on for too long is used with tubulin as well. So tubulin is itself also a GTPase, and it is in its inactive form with GDP, but in this case, active and inactive have to do with whether or not they'll form tubulin, whether or not they'll polymerize into that ultrastructural molecule, the microtubule. So if we start off with a dimer of tubulin, it's got two GDPs bound. We then can exchange those by removing the GDP and activating by adding two GTPs. This form, the active form, wants to polymerize. Once it hydrolyzes, then it will go back to this depolymerization form. So again, we're going to time how long we let polymerization go on by how easy it is to load GTP. And there are going to be other factors. We don't call them GTP exchange factors or gaps like we did with Ras, but there are other factors that control that hydrolysis rate. So we can either leave it in the polymerization form longer, or we can depolymerize by making the hydrolysis faster. So the rate of hydrolysis is going to determine how long we stay in the active form for polymerization. So here's what that looks like in the context of microtubule formation. We've got the GTP cap because we just recruited tubulin dimers to this structure, and they all, of course, have GTP because that's the polymerization form. If this stalls or we increase the rate of GTPase, then that GTP cap is going to shrink because we're going to start hydrolyzing the GTPs here, and then they're not going to want to stay in the polymerized form. Once we expose the microtubule end without GTP bound to it, like this over here, then of course this thing is going to shrink away because none of the GDP bound tubulin wants to stay in the microtubule. It'll stay in the wall of a microtubule over here because it can't get out. It's bound to too many neighbors. So GDP form is stable in the wall of a microtubule but not at its end. So if we hydrolyze the GTP quickly enough, we can make a GDP tubulin end, and that's what's going to lead to the catastrophe, the shrinking away of the microtubule. So the dynamic instability properties of tubulin are going to be determined by how quickly the GTP is hydrolyzed with faster hydrolysis, leading to more rapid catastrophe, and slower GTPase activity, leading to stable polymerization. Ultimately, that means if we want to build a structure like the mitotic spindle with something like tubulin, we have to control the rate of GTPase. If we're hydrolyzing GTP too quickly, the dynamic instability will lead to very short microtubules, and they'll never make it out here to the kinetochore where they need to be attached and stabilized. If we hydrolyze GTP too slowly, then the dynamic instability will shoot right past the kinetochore target and will allow the microtubules to be much too big to form a useful structure. So if we hydrolyze too slowly, we get things that are too long, and if we hydrolyze too quickly, we get things too short. So we have to make sure that the GTPase is adjusted for every context so that we make the kind of structure we're looking for right in the middle here. So this is why we need to be able to control the GTPase rate with various G proteins. In the case of Ras, we need to make sure that we don't leave the signal activated for too long, because that'll lead to excessive proliferation, and you can then see why a Ras mutation that blocks that GTPase activity would lead to excessive proliferation. And in the case of tubulin, we're again using it as a timer to make sure that we've got the right rate of hydrolysis, because that's going to determine what kinds of structures we can build. So the rate of GTPase activity needs to be controlled so that we can build the right structures and proliferate at the right rate.